call back a little bit to a couple of the sites that Emily mentioned before. And what's exciting for me is that I'm in San Diego for the first time, so I finally might be able to go see one of the field sites I'm about to talk about. And I had no idea that there were actually palm trees down here around my supposedly extreme field site. So that'll be a funny dichotomy there. But so yeah, we're going to be following the salt, and it's going to be a lot of salt. So hang tight there. So the first field site, South Bay Saltworks, which is down the road, is a salt farm, which as water is taken from the bay, it is brought in, and the people there are basically taking the halite out and using that to, I believe, soften the water here. And then the magnesium chloride also gets concentrated and separated. And so you get these really sodium chloride concentrated regions and magnesium chloride concentrated regions. And then you also have regions which are a lot more like seawater. And so that's South Bay Saltworks. That's one of the sites I'll be talking about a lot today. And what we're investigating there with oceans across space and time is, can biosignature molecules be detected in these sites with these super high salinities? And then how does that impact our search for life elsewhere? And then how do these vary with things like water activity and ion concentration? And then here's another analog site in Western Australia. This is Lake Campion, which is a very interesting site that we looked at. So these are transient lakes, and so they evaporate during the drier, warmer months, and then they fill up during the winter months, and so you get these salt crusts which build up on the bottom of them. And so these are really interesting because the evaporation process is happening very actively, so we can get a modern look at what's happening there. And so, in general, analog sites can give us training grounds for robotic exploration on other planets. They can help us test our instruments, and particularly we want to know how salt and acidic low pH environments affect our instruments. And then it can give us insight into the environmental processes and the biology happening there. And so now I'm going to shout out Luke Fisher's review paper in Environmental Microbiology, where he talks all about biosignature preservation in brines, particularly in deep hypersaline anoxic basins, which I won't be talking about today. But when it comes to brines themselves, as opposed to evaporites or salt crystals, we know that DNA and rRNA are well preserved, while mRNA is not as well preserved, and that ATP preservation has been observed by Tuovola et al. In 1987, but not much has been done about that since then, and lipid preservation is being studied more and more recently, and so that's good, but these aren't nearly as well explored as genetic compounds. And so one compound that we're looking at, ATP, adenosine triphosphate, it's a short-lived molecule, and it's used in astrobiology and in ecology as a marker of microbial activity. And so if you have a lot of ATP, it means that a lot of microbial activity is happening. If you don't have any microbial activity, then you're probably not going to have much ATP. But it's very highly evolved. So as a biosignature, it might be less agnostic than, for example, amino acids. And so we could look at polypeptides, or we could look at individual amino acids, and we target biological amino acids in this study, as well as osmolate amino acids, which are solute compounds that are accumulated by microbes when they're under high stress in hypersaline sites. Yeah. So, and that's well understood in the literature that these osmolates exist. And so first I'll talk about the methods that we used in the field, or near the field in the motel room. We looked for ATP using a luciferase assay. And so say you have a beautiful crystal from the crust of a lake like this from Western Australia, then you take it to your Western Australia motel room and you do you set out your samples, we weigh them out, we extract them in hot water, and then we add our luciferase and luciferin, and we have a field portable luminometer in order to do our analyses. And so that's how we quantify ATP. And moving on to what we do at Georgia Tech when we bring the samples home. So I was so glad to hear that another person here was using capillary electrophoresis, so I don't need to explain it in its entirety again, but it's a separation method that in our lab we pair with laser-induced fluorescence. And so laser-induced fluorescence means that we're tagging our amino acids, our compounds of interest, with a fluorescent dye, so we're targeting specific compounds. And so this is a targeted technique which has very low limits of detection and very high sensitivity. And so we did this at South Bay Saltworks on one of the samples that has a water activity of below 0.4 and magnesium concentration of more than 4 molar. And this is only a 1 to 10 dilution. So using a method that Marshall Seaton and I developed, we got the concentration of amino acids in this sample down to tens of nanomolar. The limits of detection for this method in general are in the tens of picomolar. And we did this using a commercial instrument. And again, it was only a 1 to 10 dilution for this hypersaline sample. So we were delighted that the results came out like this. And then for an untargeted analysis, we can use MicroCHIP capillary electrophoresis paired with high-resolution mass spectrometry. So if we take the same sample and we look at it using an untargeted method where we can get the mass-to-charge ratio of the compounds that we're interested in, then combining the zip chip commercial setup with a ThermoQ Exactive Orbitrap, this is what the chip looks like, it's very cute. We get a very interesting set of compounds which we didn't necessarily target. And so moving back to the LIF analyses for the results, what we first observed was that as we increase in magnesium concentration and decrease in water activity and increase in chaotropicity, we get a significant increase in the dissolved free primary amines that we were targeting. And so that would be our biological amino acids, which you've identified here. And as you go up, you have higher magnesium sites, and we have a lot higher peaks, and we also have a lot higher values for quantified amino acids. And so we attribute this to evapoconcentration. As the sites, which have a lot of water and a lot of salt, and then evaporate and leave the salts and the organics behind, the concentration is higher. So that's pretty straightforward and simple, but it is very useful in the case where we're looking at very low biomass regions. So next, we then put it in MicroCHIP CEMS, and we looked for these same compounds, and we found a lot of them there. And so you can see histidine, glycine, alanine, isoleucine, and leucine are resolved there, serine, et cetera, glutamic and aspartic acid. And this was only a 1 to 4 dilution of that same sample. So we were really delighted by these results as well. And in the same separation, just looking at different traces, different M over Zs, we see a couple polypeptides, and we see some adenine and guanidine in there as well. And so these are things that we didn't necessarily look for in the CELIF, but now that we're combining it with an untargeted method, we can get a look at some more compounds that we didn't necessarily expect. And then also osmolytes, which we did expect and did want to look for. We found those like ornithine, sarcosine, and betaine in our samples with very high salt stress being applied to any of the microbes that were alive there. And so these are there in significant quantity. And in Western Australia, briefly, I'll just show you some preliminary results. This is Lake Brown, which has a sodium concentration of near 5 molar and a water activity of 0.89, so not nearly as low a water activity as the South Bay Saltworks sample I just showed you, but it has high relative concentrations of a lot of these compounds, including the osmolite compound betaine, and some lower concentrations of other compounds, including a polypeptide. And then conversely, at Lake Gunter, which has a higher water activity and a lower sodium concentration, we see that there really isn't any easy way to detect those organic compounds with the method that we're using right now, so just diluting these samples. So perhaps this indicates that and of course this is only 2 data points with Western Australia, but looking at what we found at South Bay Saltworks, perhaps these saltier sites are actually places where it might be easier to detect biomolecules because of evapoconcentration. And we also want to look at the normalized abundance of certain amino acids just to see what sort of patterns we could find. And if we have water activity increasing from left to right here with the magnesium sites here, we see that certain amino acids are upregulated in this magnesium heavy site and others are downregulated. And with that information, we decided maybe we could classify our sites based on the amino acid distribution. So if you take the distribution in, you put it in a classifier, either logistic regression or random forest, you could get out one of these 3 different site types. And this is a small data set and more of a proof of concept than anything, but it's interesting to see that we have a very good area under the curve of 1 or 0.83, which, and we also can see which compounds are driving that the most. And so that's an interesting thing to take note of that might be used in the future if we have much larger data sets. And so the last thing I wanted to talk about, ATP, if you remember. So on the x-axis here is magnesium concentration, and then we have 3 y-axes, because why not? And first of all, I want to mention the cell counts by microscopy, which is from Ben Klempe, show that the number of active cells in these sites go down as you get to the super high magnesium concentration regions, which is also what Emily showed in her talk. And so what we then can look at is the ATP concentration, which strangely goes up, even as the concentration of active cells goes down and the expected activity goes down. And so we wondered, why is that happening? Because as we know, the amino acids are concentrating, but amino acids don't decay on really short timescales. They're not broken up by hydrolysis or by ATPase enzymes. But what we think is that something must be driving this, and if it's not evapoconcentration, we're wondering, since the ATP hydrolysis rate is much quicker than the evapoconcentration rate, why would the ATP accumulate? Doesn't really make much sense, but perhaps it's being preserved. So this Tuovola et al. Paper from 1987 that I mentioned way earlier, they found that ATP was preserved in low water activity sites. And so that could be preventing the enzymatic and natural hydrolysis of ATP, leaving it to persist in the solution. And additionally, it could also be used as an osmolyte compound as well and be accumulated by those microorganisms before they end up in a super high magnesium site and not very active themselves. So, in conclusion, we can detect biosignatures at micromolar concentrations in near saturation brines using CE-LIF and MicroCHIP. And amino acid distribution ratios can be well classified based on their brine type. And osmolytes are present and detectable in some but not all of the South Bay Saltworks and Western Australia brines. And we found preservation of ATP at South Bay Saltworks. And so basically, my argument is then, why don't we follow the salt and see if we can find biosignatures there on other worlds? So thanks for listening.
Thank you, Chad. We have time for 2 questions.
Chad, thank you for the talk. That was a beautiful separation that you showed earlier. So I'm curious. You did a quantitation of amino acids using CE-LIF method, and it looks like you've done some work using CEMS. Have you tried quantifying the amino acid content in the same sites using MicroCHIP CEMS and seeing what those look like to try and cross-validate both methods and using the same sample yet, or have you not got to that yet?
Nope. So we just started doing the MicroCHIP work over the past month, and so we're really happy how it's been doing with qualitative untargeted work, but we haven't gone into quantitative because the ion suppression effects might be a bit of a challenge to overcome, so that'll probably be a little bit of a longer project. What we have done to try to corroborate the amino acid concentrations is look at the expression of, or not the expression rather, but the presence of certain proteins in the genetic code of the microbes that were found to be present here by other OST collaborators. And we find a moderate correlation between the amino acids we see and the amino acids that they see, but since we don't have transcriptomes from all of these sites, we don't know exactly what proteins are being made. And so I think that's the next step is to connect those from metabolomics to transcriptomics to genomics, just as the talk last night was discussing, so yeah.
Thank you.
Any last question for Chad?
I have two questions actually.
Yeah.
Maybe I didn't really catch you. What's the pH of the side that you show the magnesium concentration is more than 4 molar?
That's a great question. I don't know off the top of my head, but I can get back to you in a moment. Yeah.
Okay. And also, how would you determine your ATP is accumulated or protected under high saline water or like low water activity?
Sorry, say again,
How do you know that your ATP is not hydrolyzed?
So, yeah, I don't know that it's not hydrolyzed necessarily, but I am predicting that. So the typical destruction method of ATP, either in the cell or in the environment, is through hydrolysis, either performed by proteins or just happening naturally in the environment with water. And so those are the things that must, from what I'm observing, have been stopped if the ATP is going to persist so long for it to be evapoconcentrated to those high of levels. The huge spike there likely wouldn't have been possible if evapoconcentration weren't a component. And so, unless the osmolyte effect is so much more significant than I had thought, that could be another reason. But I still think that the preservation of ATP is linked to the lack of a rapid breakdown mechanism.
Have you ever considered that ATP with divalent cations form some nanostructures to make the things happen?
Yeah. So whether it stabilizes it in some way, So I don't honestly know much about how I would investigate that. I've spoken to someone recently about potentially modeling it using molecular dynamics, but I haven't, waded into the actual the structural chemistry of whether that would happen. But if you have any ideas, I'd love to hear them. So, thanks. Yeah.
Thanks, Chad.